Hi everyone. Welcome to our bioCADDIE external webinar. In today's presentation, we're going to have Henning Hermjakob. He's the leader of the Proteomics Services team at the European Bioinformatics Institute. He's also a founding member and co-chair of the HUPO Proteomics Standards Initiative and member of the executive committee of the British Society for Proteome Research, contributing to the standardization of data representation in proteomics. In today's presentation, he will talk about the ProteomeXchange. And please welcome Henning Hermjakob.
Okay. Good afternoon, ladies and gentlemen, and thank you for the chance to present here. I guess I can, slides. I can control the slides myself. Yes.
Yes. We can change for you if you want.
No, okay, I got it now. It's just
okay.
There's the usual delay in the changeover, so this should be fine. Okay, so today I'm going to present ProteomeXchange, an international infrastructure for open proteomics data, and I will try to focus on aspects of interest for the bioCADDIE context, but I'll start on a much more specific level, and I'll give a 3-slide introduction to proteomics. So proteomics is about identifying and quantifying all or as many as possible of the proteins and their specific forms in a certain sample. Here, this little rodent in the picture. The sample is then run in a mass spectrometer and will produce between thousands and several million mass spectra, which is the raw data output of the mass spec machine. These are then processed, searched against databases, which is a relatively compute-intensive process to generate the output data, namely which proteins are thought to have been in the sample and in which quantities and in which specific forms. So, for example, which post-translational modifications have been observed on these proteins. And this is shown here in this little figure on the left. This is a view of one specific protein identified by its UniProt accession number. You can see the sequence, you can see which peptides, which parts of the protein sequence have been observed, also in this overview diagram, and then which modifications have been observed, and also in the summary, because this integrates over many different samples. It says then in which samples this protein has been observed. So moving more towards the data representation aspect, what are the relevant parts of the data? We want to make accessible the metadata, and this comprises the sample description, the instrumentation, and the processing approaches which have been taken to convert raw data into the results. Does everybody see if I use the mouse cursor as a pointer?
No.
Okay, then I will do this acoustically, so I will try to say where I'm pointing, but I think so far it was fairly straightforward. But I'll try to sort of comment on the pictures then. Okay, so these are the 3 major data types we are dealing with, which I'm sure all of you are familiar with, even if from a different scientific domain. And all of these we want to store within ProteomeXchange and make accessible to the scientific public. One thing I should mention is that probably in common with many other domains, we have started out with very comprehensive, complex metadata definitions. These were published, were relatively little adopted in the community because they were too complex. We basically ended up not getting the data. And one of the reasons was that people said the metadata is too complex. And so we have, for ProteomeXchange, significantly scaled back the metadata requirements. And since then, at least, we get very significant amounts of data. Okay, so moving from the overview description of what proteomics does to what are the kinds of aspects which are relevant on a more abstract level and probably of interest to this audience. Proteomics is marked by still very rapidly developing instrumentation and data processing approaches. There's a multitude of really fundamentally different workflows sort of settling down in the field, like it has happened, let's say, on Affymetrix chips, or currently Illumina sequencing has not happened yet. And there is still a lot of experimentation, innovation, but also going out of fashion of methods. This results in a lot of complex output data types in many different file formats. We have worked a lot on standards development to try to tame this a bit and rein it in. And this has worked, I would say, with medium success. But we have definitely made a lot of progress over the last years. An important point is the results of an experiment depend not only on the actual experimental setup and instrumentation, but also very significantly on the analysis approach. And that means that the same data can be meaningfully reanalyzed with a different question in mind. Data set sizes vary from probably less than 100 megabytes to our currently largest data set is around 4 terabytes. So huge variation and very significant maximum data sizes. There's not a strong tradition of open data in proteomics. Still nowadays, it has improved a lot, but we are still away from the situation in genomics or other fields where, let's say, protein structures, where it's just you make your data openly accessible, and that's a precondition to publishing. But we have made some progress. Oops, that was the wrong slide. We have just 2 months ago done a survey to find out and to really have hard numbers what percentage of all the publications in the 3 classical proteomics journals, which should have open data attached to it, actually do have open data in a public repository attached to it. And that's shown here. So the big green bars are these papers have generated data. They are data publications, but the data is not available in an independent public repository. There might be something on the author's website, but generally just the data is not available for these. And then the other, the second largest bar is ProteomeXchange, so where the data is actually available in ProteomeXchange. We don't have the comparative numbers from years ago yet, but Proteomics, the editor of Proteomics, has over a few years given us kind of anecdotal numbers, and these stayed for years at about 13% of the numbers for the same question like here. So definitely with the introduction of ProteomeXchange, we have made a big step forward, but there's still also a long way to go. There have been lots of even high-level calls to make the data accessible. A kind of peak was around 2010 or building up to 2010, and almost everybody agrees, yes, data should be publicly accessible, but just in practice, the implementation has not always been perfect. But even if you are willing to do your best and to make the data which accompanies your publication accessible, there were a number of questions in around 2010 when we started ProteomeXchange, which we have often been asked. And these questions are about submission. So which repository should I submit to? There were more than one, and they were not coordinated. Should I submit to all of them? What kind of data do I need to submit? The raw data only, the processed data or results only, the metadata? And then there are questions about the deposited data. How do I find all data sets on chromatin? Do Pride and Peptide Atlas, two of the databases, both have this data set? Do they interpret it differently? And we have specifically been plagued by two major shutdowns of repositories in the domain, in proteomics, which has really thrown us back in terms of data availability by years, I think. And these are the fact that Peptidome, which was operated by NCBI, so a serious provider, was closed down in 2011. That happened in a controlled manner, so they announced it beforehand, and the data is still, in principle, available from the NCBI FTP server, but nevertheless, significant repository was just shut down because it ran out of funding. Much worse was the situation for Tranche, a quite heavily used repository in the domain, which first became unstable and more unstable for about a year, and then closed down in January 2013. And so we have been asked, okay, so if I go to all this trouble and make my data accessible, will it remain in the public domain, or will it be just vanishing like it happened to what I did last year and submitted to Tranche? So this is the background, or these were background questions which led to the development of ProteomeXchange. And this is the high-level overview of how ProteomeXchange works. So it is a federated repository. We are not replicating data between different sites. The data is only kept at one site. Only the metadata is centralized. From a user perspective, the user chooses to submit his or her data to one site, and we request all the three major data types which are essential, namely the metadata, the raw data, and the results as part of the submission. And the submission is then processed by one of the ProteomeXchange partners. Currently, the partners are the Pride database at the EBI here in Cambridge, UK, the PASL database which is part of Peptide Atlas at the Institute for Systems Biology in Seattle in the US, led by Eric Deutsch, and the massive database which has recently joined based at UCSD under the leadership of Nuno Bandeira. And these three, and potentially in the future others, each of these is managing the submissions they receive, keep them confidential until the publication which the data supports is released, and then obviously the data is made accessible, and a relatively short XML formatted metadata message is sent to Proteome Central, the site which is operated at ISB, and this site, Proteome Central, also archives all these metadata messages and makes them centrally searchable. So you can either subscribe to an RSS feed or to a Google group and get notification of new relevant data sets for you, or you can search based on the metadata for them at Proteome Central. And then you can trigger actions according to your interest in the field. So you might be a database like GPMDB, which reprocesses raw data in a coordinated pipeline and then presents their own view of this data, similar to what Peptide Atlas does. You might be interested only in a few selected data sets for a much more scientific focus. For example, our partner site COPA-KB at UCLA are a specialized resource for cardiovascular proteomics, so they pick up only data sets which fit into that search scheme. You might be a site which is only interested into the biological results, say for systems biology, modeling. You're not interested in the actual supporting proteomics data, the mass spectra at all. You just want to see which proteins have been observed in which quantitations in the system. You might be interested only in a relatively high abstraction level along the same lines like UniProt and NextProt. They provide these proteins, let's say these specific isoforms have been confirmed by mass spectrometry. These modifications have been seen, as opposed to focusing on the underlying mass spec evidence. So this is the broad setup of ProteomeXchange. As I mentioned earlier, only the metadata is centralized. The raw data, which is vastly larger than the metadata, of course, is kept in only one of the repositories. Of course, everybody is free to copy it, but we are not duplicating or replicating the data to all sites, because the volumes involved are large and the numbers of labs who are actually using the raw data around the world are relatively limited. So we might be copying a lot of data which is accessed only by a few people. But of course, each of the sites has their own backup strategies for data security. What does it really look like? When you use Proteome Central, you can search based on a relatively limited amount of search parameters, and then you get a view like the one shown here. The identifier, the dataset, the title, which repository, short sample description, instrumentation, the supporting publication, submitter, and something which has been introduced more recently on request, the lab had, and this has actually worked quite well. It was something we didn't think about in the beginning, but many people requested, and it has worked out well in talking to lab heads, saying, your colleagues, Ebersold and Mann, the leading scientists in the field, are submitting to ProteomeXchange, but we don't see anything from your lab, so what about you? So this turned out to be a good way to group and to access data sets from a specific lab, but also really to encourage PIs, to encourage their PhDs and postdocs to make the data accessible through ProteomeXchange. On the topic of easy access to subsets of the data, we are in the process of introducing so-called tags, which are simple, like you see here, tags to group different data sets which might be of interest to sub-communities, like people might be interested only in technical data sets to do, say, test runs of mass spectrometry software, or they want to explicitly exclude these and they want to only use real biological or biomedical data sets. And then the tags also allow us to group larger groups of projects, often called, let's say, the International Human Proteome Project, and group the output from multiple PIs into a larger context. This is not yet directly accessible in ProteomeXchange, but will be soon. It's already accessible on the PRIDE site in the PRIDE archive. I mentioned the lab head already, so I'm going to skip this. So when you then select data sets of interest, you get a view like this. So you see it's a relatively lightweight metadata description. The parameters I've already mentioned, a bit on the version history, the supporting publication or publications, keywords, and then you get the link where you can access the full detailed data set. And that's basically the essence of ProteomeXchange. The next slide shows how it has worked so far, and I've already mentioned we have significantly increased. We have grown not only with the field, but we have grown significantly faster than the field. We have really increased the percentage of data, of published data, which is also accessible in public repositories. And I think this is a major success. And this slide shows a bit the growth. So we started in 2012 with 102 data sets and went within 2 years to just under 1,000 data sets in end of last year, with broad contribution from lots of different countries, from lots of different sample types, and across a broad range of data volumes per study. I mentioned this can be in the low megabytes range to more than 5 terabytes. At more or less any given time point, about half of all the data sets are publicly accessible because of the time lag between data deposition and release of the data, which is a fairly normal process we observe also for other databases which provide this grace period and keep the data private in this period. So a bit on the will my data still be there in 5 years, part of the question and a few of the other questions I mentioned earlier. So clearly all databases depend on continued funding. There have been these two cases where key repositories in proteomics ceased operations. And what we have learned from that for ProteomeXchange is part of the commitment to ProteomeXchange is a strong commitment to mutual backup in case of funding loss. And in this case, the remaining repositories will do their best to safeguard and keep easily, in a structured manner, accessible all the data from repository which might cease operations. But of course, we also try to be selective in who's participating. Both Pride and Peptide Atlas have been around since 2005. And of course, we have only the past as a good predictor for the future, but that should be a good indicator. And we have already shown that we try our best to safeguard data. Pride has imported all the data from NCBI Peptidome, and it is now fully searchable within Pride. And Massive has imported all the recoverable data from the tranche failure. Unfortunately, a lot of the data sets were corrupted, so there has been significant actual data loss, and we have to try and do our best to avoid that in the future. Okay, now I'll move on to a few more specific aspects which might be relevant for this community. We have struggled for quite a long time with the complexity of different workflows in ProteomeXchange, and that means that we had to cater for lots of different file formats. We sometimes had to turn people away because we said, sorry, we can't pass your output formats. That means we can't integrate it into our databases. And that was clearly an unsatisfying situation. And so we defined two submission types: complete submissions, where we can process the result files. They can be converted to PSI standard formats and can be made accessible to the community, fully parsable, fully searchable, and, well, as a good basis for reuse. The other submission type is partial submissions, where we can essentially take any type of data, even very innovative workflows which have only just been implemented by this one lab, where essentially it is a collection of files, plus it still has to have not only raw data, but it still has to have the three components: raw data, metadata, and result files. But we are accepting result files which are not parsable with our current parsers. So the data is only really accessible through the metadata, but might still be very important for others for reprocessing, validation of publications, etc. The difference between the two is a bit shown here. Complete submissions can be turned into these integrative views I showed in the beginning, where they provide additional facts about specific proteins or contribute to these. You can browse the data in detail across multiple dimensions, different processing stages, while partial submissions are essentially a collection of files you are pointed to from Proteome Central. A key problem for us for years was FTP transfer. So if we received large submissions, we got constant complaints from users. We had to do a lot of support helping users because we provided nice tools to prepare the submission, but then there was the actual submission process, which meant FTP of large files taking often several days, with the associated failure rates and interrupted transmissions, partial submissions, etc. In the second half of last year, we managed to integrate Aspera into all the different ways of transferring the data. Aspera is a commercial file transfer protocol, basically an FTP replacement. The client software, so what our submitters or also downloaders use, is free of license fees. The license fee needs to be paid only by the server, and so by the EBI. And this has brought an enormous advantage for us. The transfer is somewhere between 10 and 50 times faster than FTP. And for us, it has made really a world of a difference in terms of much higher user satisfaction, and we get much more positive feedback. I'm a very big fan of open source software. I don't like to advertise a commercial tool, but in this case, it really has made a big difference for us. Now, is it actually used? So far I only spoke about the data in aspect. So is there actually any data out? And I've had pre-discussions in the bioCADDIE context that there is also a strong interest in usage statistics, what is actually the impact of databases. And so in 2013, or I spoke about 2013 as the rise of public proteomics data availability, and I'm very happy to talk about 2014 as the rise of proteomics data reuse. So our top downloaded data sets, there were actually 2 high impact Nature publications, and data associated with these 2 had very high download numbers, but also one MCP paper. And so these are really, these numbers are generated as total number of files from this particular ProteomeXchange accession number from this data set downloaded, divided by the number of files in this data set. So that means really this is complete downloads. If people only did a spot check and downloaded a couple of files, then this is even an underestimate. But of course, there's also a long tail of data sets which are probably accessed never or once or so. A bit more on reuse. I've already mentioned this one on the previous slide. The first one, GPMDB reprocesses systematically data sets from the public domain and comments on them. 15 of the 20 data sets of the week in 2014 from GPMDB were based on ProteomeXchange. GPMDB is not a ProteomeXchange partner, although of course we are in communication with them. And another interesting aspect is that recently there was a publication in Nature Biotechnology about a tool, Peptide Shaker, which allows to directly access data in Pride, only in Pride at the moment, download this, research, reprocess the data with new parameters, and then in principle also directly upload the data, assuming that interesting discoveries have been made which lead to a publication. In total, the Pride download volume, this is only the Pride part of ProteomeXchange, that's where I have the data for, was 150 terabytes in 2014. And I mentioned already the most popular data sets were downloaded more than 1,000 times. The role of DOIs. So many or mainly editors on the ProteomeXchange stakeholder group suggested that we assign DOIs for ProteomeXchange datasets also as an incentive for people to get them a citable unit, a citable thing for the work of submitting their data. We implemented this only for complete data sets to have a kind of incentive for authors to generate complete data sets and not leave it with the easier to do partial submissions. The implementation, so generating DOIs, managing them, was all relatively straightforward. The usage so far I would call almost irrelevant. I just got the DOI resolution report for last month. The top ranking PXD had 26 resolutions. So that means 26 times over the last 6 months, somebody clicked on dx.doi.satata PXD 1677. So compared to the usage numbers I mentioned earlier, this is not very impressive. We see similar effects on we did similar counts on the PDB side, at least PDB usage at the EBI, which resulted also in a big imbalance. The PDB accession numbers are much more used than the DOIs which are assigned to the same entry, and are, I guess, just because they are much more cited also, much more visible in papers and so on. But so the overall resolution numbers are very low. We have used a very low-tech approach for trying to make PXD accession numbers accessible through PubMed, and we are simply asking submitters, can you please include the PXD in the abstract? And this works reasonably well. So there's probably something like a third of the now publicly accessible data sets, a quarter or so, or a third, which you can find by just searching for PXD 00 star in PubMed. So this is an interesting additional way to look. And this helps us also to identify the case where a paper has been made public, but nobody told us, and so the data set would still be access protected. And so we regularly scan for this and check for new PXD numbers in PubMed, and then check if we have made these actually publicly accessible. One thing which worked very well is we are asking submitters to include in the text, again, the same thing, to refer to the data set with a PXD accession number, and we ask them also to cite that this has been submitted to the ProteomeXchange consortium. And this has been, I would say, a great success. We have currently, as of yesterday, 528 Google citations, Google Scholar citations for the Pride 2013 NAR database paper. I recently checked, I didn't do it very recently, but when I checked a bit ago, this was the highest, among the highest cited papers of the EBI, for which are so young and only eclipsed by the very large databases Ensemble and Uniprot. Okay, I think we also published a kind of instructions manual how to submit to ProteomeXchange, just to have it easy and have the whole process outlined for users. And now I'll move on to the last part, the perspectives. This was where we are right now with ProteomeXchange, and a future perspective is what I'm going to outline in the next 4 slides. There's a kind of sister organization to ProteomeXchange, BioModels, which has been in part and is still in part being modeled on ProteomeXchange. And BioModels has a similar overall consortium collaboration architecture, and we are in good communication with them. Another resource which has a different character is the European Genome Phenome Archive, or EGA. This is an archive for access-protected data, which is private and is only accessible to selected scientists, because this contains human, individually identifiable data. And the approach EGA takes is they make the study metadata accessible, and you can then identify based on the metadata which studies are relevant for you. And you can then ask the corresponding ethics board for access to the data, and after a relatively lengthy process, if you're approved, if your research project is approved, you then get permission to access this data and analyze it then as you have outlined in your research proposal. And what we are planning is to integrate these two into a multi-omics data discovery index. So we would develop an infrastructure for integrated data discovery across ProteomeXchange, BioModels, and the EGA. The technical challenges are there, but should be feasible. The real challenge is the common metadata representation across these fairly different omics repositories. But if we succeed, then we have a demonstrator for findability across omics, which would involve 8 repositories in 2 continents and 6 organizations. It would have the interesting aspect of findability across open and controlled access repositories. And it would be a kind of pilot for collaboration between BD2K and EU Elixir, and hopefully we will be able to coordinate this with bioCADDIE, because this clearly is something which was planned already in our BD2K cardiovascular project, and we have to do this anyway to fulfill our grant obligations. But after the fact, we discovered that there is a strong overlap with bioCADDIE, of which at least I wasn't aware when we wrote the grant. So I very much hope that we can coordinate efforts there to make this in a good coordinated way. And with that, I'd like to acknowledge the many people who contributed to ProteomeXchange, PIs at the different sites, the PRIDE team, in particular Juan Antonio Vizcaino, who is the PRIDE project lead and who has done a lot of the work which I've been presenting here. and coordinated a lot of the work. Rui Wang, Florian, Attila, and Tobias. Attila and Tobias are the two curators on the Pride side for ProteomeXchange, which I think are essential for the success. They have really built up very good submitter relationships. We have had good support from editors, although submission is still not mandatory, but we are at least gradually working towards that. And of course, the funders and all data providers without whom we would have a rather boring, empty database. Thank you.
The conference has been unmuted.
Okay, so we unmuted all lines, so now the webinar is open for questions.
Hi.
Go ahead.
Go ahead.
Yeah?
Sorry, I started asking a question at the same time as somebody else did. Let that other person go first.
I can wait.
Oh, okay, so this is Ian Four, and I'm the science officer on the bioCADDIE project. And first of all, Henning, thanks for recognizing that connection. And I think it was talked about in the EU BD2K Elixir discussion that we had in January, and this provides a lot of useful detail behind it. I don't think that certainly wouldn't attempt to do this here, but I thought your slides that where you colored up the different kinds of data are going to be a great example that we can look at to understand where we're talking about different kinds of metadata. And as we look at that pilot, it will be useful to explore that. I did have a more trivial question, but that can wait. I'm finished.
Okay. So should I read out the chat questions? There were a number. I don't know if they are visible to everybody. Should I read them and answer them, or?
Yes, you can go ahead through the questions in the chat.
Okay, so there was a question from Arkad Jasekar. Are there plans to replicate the submitted datasets for fault tolerance and crash recovery? So, as I mentioned during the talk, we are not doing this systematically because we trust that each of the organizations have a valid crash recovery policy. All of these organizations are high-profile bioinformatics organizations, and frankly, if they fail to a level where we wouldn't be able to get the data anymore, then there's a lot of other databases also hit. So just as an example, EBI has compute centers and data centers at 3 physically different sites and has backup strategies across these 3 different sites, which are not relying on tapes, but which are relying on spinning disks, because we calculated at some point if we had to restore the EBI from tapes, we would need about 6 months. And so in a similar vein, each of the involved organizations have to have their own strategy. That's also why we want to be fairly selective about who is joining the ProteomeXchange consortium to ensure that there is really institutional backing as opposed to an enthusiastic PI or postdoc investing the time but then not being able to maintain that. But nevertheless, of course, there might be a systematic duplication in the future, something which we have discussed already. As an example is Chinese ProteomeXchange node, and there the problem is simply that the transfer across the Great Chinese Firewall is extremely slow, and it would make no sense to transfer the same data set across the firewall multiple times. So we would implement at least some kind of local caching, if not replication, simply for efficiency of access. Did that answer the question?
Yeah, thank you. Thank you very much.
Okay, the next. Have you considered including DOIs in the references that you ask authors to include in their abstracts? No, we haven't, because as far as I know, I rarely see DOIs included in abstracts, because I think normally the overall paper has the DOI. This would happen normally somewhere in the text. And we wanted to minimize, let's say, the ugliness of the statement. And the PXD simply looks less ugly than the full DOI URL, which would need to be included. So that is the thinking behind this. That's why we didn't do this. Also, yeah, I think that's the main reason. Are there plans to coordinate with Geo or Array Express with respect to transcriptomics datasets? No. So they have their own partial, as far as I know, data exchange, and of course they are much bigger just in terms of volume. So we are not planning to coordinate at least the core data with either of these. Something which would be interesting in the long run, of course, is to integrate on the metadata exchange with Geo and Array Express. But this is, I would say, after the first pilot with a suitably broad number, but not of gigantic repositories. Okay, so some of the metadata, sorry, did that answer the question? Can I just always ask for a short confirmation that that's...
Yes, it does. Thank you.
Okay, so Kim, some of the metadata that source data repositories collect contain free text. Are you processing them further to the ProteomeXchange side to tease out key information in a more structured way to facilitate data search and integration? So no, we are not processing the data we get from the submitters. Until taking the next question, we are using, obviously, the NCBI taxonomy. We are, at the moment, not even using the controlled vocabulary for instrumentation, which we have. And this is based, again, on the strong pushback we got before. So we have been really frustrated by our very orderly, let's say, approaches we followed in the beginning, where people said, no, we don't want to have endless drop-down menus. And of course, then we said, okay, we have auto-completion. Yeah, but I don't know which is my machine. So now we are giving the users more freedom. We have, of course, less structured data, but at least we get the data. So I think over time we will gradually swing again in the other direction of trying to achieve a more structured way. But for now, we said, okay, we are happy with the rapid increase in data. I think it's still very searchable, very approachable, but yes, over time, probably also in step with increasing journal requirements, we will be able to again demand more structured metadata. But at the moment, at least, we have a high level of user satisfaction, which we have never enjoyed before. Let's put it like this. No, it's true. I mean, I'm talking really from long experience, and it has really changed from in terms of user feedback. It has really changed from frustrating business to something which it's now quite nice to see the user emails at the end of a submission process or so. And we came into this from a very different perspective, and I think we have learned something. Besides mass spec, are there other significant platforms in proteomics, for example, protein sequencing? That's again EN4. There is other proteomics platforms. The foremost one is probably based on antibodies. There are, and more frequently, other alternative antibody formats. There's a large, very valuable resource, the Human Protein Atlas by Matthias Uhlen, which provides data which is very similar in the sense it is a large-scale resource and protein expression data. We are not at the moment planning to include this, but we have actually had brief chat with one company who provides one of the alternative antibody platforms who were interested in having a public repository for their research results. So in principle, I think this would be very interesting to do. There are no immediate plans for it, but as we are already planning to extend into a multi-omics data discovery index, I think it should be perfectly possible to do the probably easier inclusion of other proteomics strategies. Okay, Anu, how granular is the search? Does the results return data sets, or can one search for specific proteins and peptides? So the search is only based on metadata, so you can only find the relevant data sets. And as I mentioned, for some of them, the data on which proteins and peptides have been identified and quantified is not even available because it's in formats which are not parsable. Even for those which are parsable, this is not part of ProteomeXchange. But we are already working on the next step, which is kind of building on ProteomeXchange, and that is an API for making the actual data computationally accessible. So the idea is you can first zoom in on the relevant data sets, and then you could actually query specific data sets at the level of protein, peptide identifications and quantitations, but that is still very early work. But there are considerations. At the moment, the answer is no, but we are planning this at a different level, not directly as part of ProteomeXchange or the data discovery index, but as a next step. Any hints to, okay, Philippe Rocacera, any hints as to solving the terminology problems? Which options are being explored? So at the moment, we are not exploring any options actively, but there are discussions with SEDA, one of the other BD2K consortia, to see what we can do to do exactly what was hinted at here earlier, which is not ask more of the submitters, but to generate more structured data through post-processing. So this is at the moment still at the level of early discussion. Okay, Huo Xu, does ProteomeXchange provide API web service for external queries from a third party? ProteomeXchange at the moment does not. This is something which is on the to-do list, and this is also something which Pride and I think also the other partners at the moment already implement. So I think this will be added soon, but it's not yet.
Okay, thank you. I think we reached our time today for this presentation. So I would like to thank you, Henning, for this great presentation, and thank you everybody for attending our webinar. And the next webinar is scheduled for April 9. So thank you, everyone.
Thank you.
Bye.
Thank you, Henning.
Thanks.